Hello, everyone. Thank you all for joining ChiroSolve's first webinar. This will be followed by many more in the coming year. We felt that as the first webinar on the complex topic of chiral resolution, it would be best to start with an overview of the available technologies by the experts who have been offering the tools and services for chiral resolution for many years. Before we move forward, some housekeeping announcements. We will be recording this webinar and we'll make it available in part per presenter on the ChiroSolve's website. So if you do not wish to participate in the recording, please exit now and you could enjoy the recorded webinar when it is available. We request that if you have any questions, please post them on the chat window with the name of the speaker it is intended for, and we will collect them and get to answer as many as those as we can after the presentation. As you all know, more than 55% of pharmaceutical drug molecules and many used in agrochemical industry are chiral in nature, many of them with multiple chiral centers and many enantiomers. In recent years, it's become mandatory to study each enantiomer before you can bring a chiral product into the market. As such, chiral separation is an important topic for pharma and agrochem R&D. Today, I'm very excited to introduce to you 4 industry experts who have been solving some of the most challenging chiral resolution problems for their clients in pharmaceutical and agrochemical industries. They will share their unique experiences and may discuss some of their challenging projects with you all. Our first speaker today is Dr. Nitin Vaidya, who is the founder of ChiroSolve and the creator of the ChiroSolve screening kits. ChiroSolve offers chiral resolution products and services based on stereomeric crystallization. During his career of over 30 years, Nitin has held many technical and executive positions related to drug discovery and process development. Our second speaker, Dr. Paul Lefebvre, is the general manager at Averica, a neo-pharm labs company offering full-service chromatography CRO services, including purification by SFC and HPLC. Paul has worked in separations and purifications for over 25 years and has a deep expertise in purification. Before Averica, he worked at Waters Corporation, developing an advanced mass-directed purification. Our third speaker is Dr. Edward Franklin, who has over 15 years of experience in doing fundamental investigations of kinetic performance of packed bed HPLC columns, instrument and column development for high ultra-pressure separations of complex samples, and column stationary phase development for application-specific methods. As the technical director of chromatography applications at Regis Technologies, Inc., he develops new products and methods for chromatography solutions related to chiral and achiral separations. Our fourth speaker today is Gary Yannick, who has many skills including physics, mathematics, optics, lasers, electronics, mechanics, and software. Utilizing this prolific background, he has been developing and manufacturing laser polarimeters and offering software for automating method development and purification. These technologies have been in constant use at many locations for rapid chiral purifications. Dr. Yannick's application experience includes HPLC, UHPLC, SFC, CCC, CPC, SMB, and PAT. So without further delay, let's get to our first speaker, Dr. Nitin Vaidya, who will discuss some of the challenging chiral resolution projects and how he utilized the available resolution technologies to deliver them successfully. Dr. Vaidya.
Hi, Nitin. Can you just change the view?
I see that presentation view. It is presentation view.
Yeah, you can just swap it. Yeah, looks good.
Okay. Right.
Can you see it good?
Yes, yeah.
Okay. Okay. Good. Okay. Thanks everybody for joining our first presentation and first webinar in the series. My name is Dr. Nitin Vaidya. I'm a tech CTO of ChiroSolve, and everyone, thank you for joining. ChiroSolve specializes in diastolic crystallization, and since 2005, we started this company. 2005 has been offering product and services, and that helps you use technology most effectively. Today, I will offer a quick introduction to our product and services and discuss some of the challenging resolution we have performed. ChiroSolve kit have been used by pharmaceutical and agrochemical companies during early R&D by both drug discovery and process development groups, as well as specialty chemicals. The kits offer 384 resolving agents and solvent combination within a single package that provide result within 24 hours. All the resolving agents and solvents we use are easily available in market, large quantities. As a result, you get conditions that are scalable. Our customers have used the kits for following points: to define pattern scope, to identify possible resolution conditions to cover in their IP, to obtain higher purity when other chiral separation methods cannot achieve. Many times other chiral resolution and chiral synthesis as well as enzymatic resolution get stuck with particular percentage of purity, and to get final purification, you may have to use diastolic crystallization. Finally, during the early method development to identify most feasible resolution options during pilot, the following 2 points. During pilot studies, proof of concept has stage to get small quantity of enantiomer. The kits offer very unique advantages: quick and consistent results. You know most of the commercially viable resolution conditions within 1 day, that if it done manually might take as long as 1 month. So it really advantage over there. Elimination of human errors through full automation. Use of the kits can be easily automated as possibly you can, identifying feasibility of chiral resolution for all identified targets in one sitting. Exhaustive screening. We offer 384 commercially viable resolution condition of both positive and negative isomer within one sitting. So instead of doing tedious work one at a time, we are able to do it simultaneously. That's why result can be within 24 hours of times given. And also we have this long shelf life. Kits are made of chemically inert substances withstand extreme temperature and reagent solvent provided in separate plates to ensure long shelf life. ChiroSolve offers chiral resolution services where we use our kits to identify the best resolution candidates and then identify the ideal resolution methods using the best resolution techniques. The services are following. Pure isomer delivery takes 1 week, and we deliver up to 5 grams of isomer. Here the goal is to provide isomer of target purity as fast as possible using any resolution method needed. We use chromatography, SFC, and/or diastolic crystallization, or combination of these methods. Which technologies is to use is depend on complexity of the racemate, number of chiral centers, and number of isomer needed for derivatization or blocking of the functional group. And the speed and amount of isomer needed by the client. Method development for scale-up takes about 2 weeks, and we deliver up to 50 grams of isomer. Goal of this service is to identify the optimal separation process that yields maximum amount of enantiomer with target purity using minimum number of recrystallization steps while minimizing amount of solvent needed. And here are some of the examples which we encountered during our 20 years plus company which is going on. One of the compound we were dealing with the Spiro compound, where 3 of the drug discovery companies came to us to resolve their chiral Spiro molecules. And clients specifically needed scalable methods. They wanted to like a kilo lab and above. So we decided to pursue diastereomeric crystallization. And the first compound we were dealing with carboethoxy ester and carbotubutyl ester, which was a neutral compound. Being neutral, we customized the strong acid kits with several sulfonic acids for screening and pursued 3 optimal conditions to define the resolution. And we're successfully able to provide 6 grams of 96% PE isomer along with 3-step process. Second compound was monoester. It was slightly basic, therefore common acid could be used for resolution. We didn't have to go to strong acids. And 2-step resolution method was developed for large scale adoption. And the third compound was dicarboxylic acid with benzyl protected cyclic amine and had a solubility issues. To circumvent this problem, we used protecting group carbotubutoxy ester. The resulting compound was then resolved using SFC for fast delivery of initial batch. Full method development was then defined using our base kits. Second example was a pyraphthrins. As a pyraphtrin is been is used in agrochemical, and it is used as insecticide, and it is very popular among the application process because of it's been not very harmful to human beings. So it is very used effectively as an insecticide spray. And this pyrathrin typically has a cyclopropane moiety. There are 2 fragments, chiral fragment can be. One is a cyclopropane fragment, other is a alcohol fragment or amine fragment is there. And most of these molecules are, as I said, cyclopropane with bunch of chiral centers. And typically there are 1 to 3 chiral centers. And as you know, the rule by rule, 1 chiral center yields 2 isomers, 2 yields 4 isomers, and 3 chiral center have 8 isomers. So we especially dealt with 2 big projects, purification of biphenthrin and cypermethrin, and we did have another 4 projects also, but representation is given in these particular 2 projects. And here we use selection of base resolving agents we use based on the literature precedents to resolve cyclopropane carboxylic acid. After identifying the possible resolution condition, we try to resolve, but during coupling reaction, because there was involved esterification where you combine chirally pure cyclopropane carboxylic acid with the alcohol fragment or amine fragment, and we all noticed some racemization was occurring. So we decided to use SFC through our partner on partially resolved racemate to get the final isomer, 98% EE, and able to deliver 2 desired stereoisomers for biphenthrin and 6 out of 8 isomers for cypermethrin. So our customers were very happy to get 6 out of 8 very pure stuff in case of cypermethrin. The other process was the scale-up issues, because especially one of the company, generic manufacturer for duloxetine, were approached to us to install. They wanted to find out whether diastereomeric crystallization could be used to scale it up for duloxetine, and can add their portfolio in IP. And we took this molecule and went through our screening, and we identified a number of resolution conditions. Especially 5 of them were identified pretty efficiently. One is mandelic acid, 2 methoxy, 2 phenylacetic acid, 2 phenylpropionic acid, tartaric acid, and L-ditolulyl tartaric acid. And out of this, mandelic acid and ditolulyl tartaric acid were the most suitable and effective resolving condition reagents. Finally, we installed mandelic acid based on several factors, because ditolulyl tartaric acid, even the resolution efficiency was looking very efficiently and comparable, was still expensive and difficult to recover and recycle. So we selected mandelic acid for two main reasons. For number of crystallization, we had to use one less crystallization to get high EE, and ratio of racemate and dissolving agent 1 to 0.5 was used in the process. The next batch of compound, we are dealing with boronic acid. The boronic acid is getting very high efficient therapeutic targets. It's very popular among a number of companies, including startup companies as well as pharma companies. And we were dealing with one project that initially they did some boronic acid compound, and after boronic acid, which was a free amino group and boronic acid group, they had to protect the amino group after making a free amine with a Bock protection using Bock anhydride. After the Bock protected amino group was installed, use the chiral chromatography, they were able to separate and make a large quantity of this N-Bock compound, boronic acid complex. But again, you have to add extra step, for deep protection of the Bock. So if you basically had to add two steps, one to add and one to remove, So then we got the project, which was an analog of boronic acid, and which also was a sort of internal salt. So we use ion exchange resin to deep sort of remove which was a closed structure, and remove it. So the amine was free. And the amine, when the amine was free, we thought we might have to Bock again. We had to protect with the Bock. But lucky, simultaneously, we tried directly our kits on the free amine. And it was a good break for us, and we were able to resolve the free amine with the mandelic acid in methanol straight. And really that gave us an advantage, reducing number of steps, especially a Bock protecting group addition and the removal of Bock protecting, removal of protection. It's just like a green chemistry, where if you remove number of steps, it's better for the process, for the kilo lab and above. And the last but not the least, the example is the neutral alcohols. There are a number of times you face the resolution work for the neutral alcohols, aldehydes, ketones, which are neutral in nature. And as you know, diastolic crystallization is basically you need to have acid or basic functionality to be able to resolve with the acidity, acid kits or basic kits. And here we have this, especially this alcohol, which was 1-phenyl, 1-propanol, CAS number that given over there. And so we have to resolve this alcohol, but you cannot resolve because it's very neutral. So we have to, first of all, we have to either get install carboxylic acid functionality there, or succinic, or either acidic or basic. So in this time, for this particular alcohol, you tried phthalic anhydride as well as succinic anhydride to install free carboxylate. But in our hands, we did not have good success with phthalic anhydride. So this particular case, we stick with succinic anhydride, and we able to prepare this compound with the dichloromethane and triethylamine with the succinic anhydride to get monosuccinate. And monosuccinate compound, then we able to use our number of basic resolving agent and identify 95% ethanol in cinchonidine to get good results. And this was further established in the kilo lab and further up. That was my last example. Thanks for joining our webinar. We'll give it to